Hi, Jason.
Hi, how are you?
I'm great. I'm great. I'm so glad that you can join, and thank you so much. I really appreciate it. Um, we have been having these career conversations with people who are doing really interesting things in synthetic biology and biological engineering, and you are one of them. Plus, you are a big member of the BioBuilder community already. We got to see each other virtually yesterday at the BioBuilder Club final assembly. Maybe we can start there. Do you want to start by talking about your BioBuilder Club team that you've been shepherding?
Yeah, so our BioBuilder Club team, they're from Indonesia, and they've been working primarily on plastic degradation. And so they've been coming up with solutions to this. And I think there's been a lot of BioBuilder interests in plastic remediation and plastic degradation. But I think they have a unique sort of take on this because I think they're really trying to develop strategies that they can implement in their community. And they actually have funding from the government to do this, as well as sort of a lot of buy-in from sort of stakeholders in the area to be able to find these solutions. And I think they've come up with some great ones. So Rania is studying how a specific fungus, I think it's oyster, basically one of the strains of oyster mushroom, can grow on different plastic materials. And so she's actually gone through and cultured this oyster mushroom on top of plastic samples and watched it grow, and basically then went through and did tensile testing to actually see, did the plastic end up becoming more degraded over time. And so she did that study, I think, about a year ago. And then unfortunately, the pandemic shut down all of her research abilities to be able to go in and do that. So we created a strategy of doing some bioinformatics on this mushroom. So we've gone through, and she found the genomic sequence of the mushroom, right? And she's put forth a hypothesis of a couple different enzyme classes that might be involved in the degradation of this plastic material, including a lacase enzyme. And then she was able to go through and find other lacase enzymes by looking at a database of different carbohydrate active enzymes. And then she could sort of match those up with her candidate enzymes. And so in this way, right, she's putting forth a hypothesis of this might be an enzyme that's expressed by her fungal cells, right? Maybe she's discovering a lacase enzyme that has slightly altered specificity or slightly altered activity towards these plastic sort of substrates. So in that way, I think she's really advancing the field. She has a goal of trying to do some transcriptomics or some proteomics with this, where she actually will go through and sort of reculture her fungus and then look at sort of underlying phenotypic changes. We've tied it a little bit to synthetic biology by saying she could then take these enzymes and make them in sort of maybe a more genetically tractable host, maybe E. coli or maybe Saccharomyces cerevisiae, right? To be able to maybe, make more of this enzyme to be able to promote degradation. So I think that's a really exciting project, and she drives all of it. I just give her a couple papers here and there.
Okay, I don't know what you were like as a high school student. What you have just described is not what I was like as a high school student, and she is remarkable. But your mentorship of that project is absolutely invaluable, and so thank you for helping.
And I'll give you a sense of what Chris is doing. So Chris is another student who has his own BioBuilder project. And he's looking at sort of how we can use plastic degradation in microbial fuel cells. And so he's planning to use a bacteria that was isolated from a plastic recycling plant a few years ago that made huge waves in the scientific community for its ability to degrade PET plastic. And he was looking at sort of just doing a single culture of this organism. But once we started looking into it, it seems like that organism might not be very good at generating electrons. So he's trying to maybe make a coupled community fuel cell where he sort of uses the one bacteria to degrade the plastic into an intermediate that can then be consumed by another bacterium who's very good at making electrons. So in that way, again, with the idea to really implement these. So I think they've been very great at doing these experiments, and it's too bad that they haven't been at the bench this year, but I think we've been able to do enough on the computer to at least get them some hypotheses to move forward with.
It's incredible. I mean, I think the resilience of the students, both to the changing plan of not being able to do experiments, but rather now focus on things that they can do, and also to pivot the work that they're doing as they make the discoveries that they're doing, it's really so mature and so sophisticated. It's skills that will carry them forward, no doubt, as they move into careers in science and engineering.
Absolutely.
And I'll say it again, these are not high school students like I remember my high school community. They're incredible, and your ability to help them along is so important, to take these students that are so excited and so creative, and eager to be able to give them next steps and awareness of what is possible and what is not. So maybe you were exactly a high school student exactly like that. I don't know. Do you want to say where you started? Were you as interested in science?
I did start in high school. So I was very lucky to grow up in Pennsylvania, and we had this competition called the Pennsylvania Junior Academy of Science. And I started doing projects for that when I was in 7th grade. And these were science fair projects that we would do, and we would pair ourselves up with teachers in school. But then the nice thing is we would go to the sort of county level competition. We would give a sort of 10-minute presentation. At that point, I think I had transparency slides that were all numbered, and I could put them on and sort of go through my presentation. Maybe I'm dating myself a little bit there. But then I was able to do this throughout high school. And then, if we scored well enough on our presentations there, we got to go to Penn State for a long weekend and then present everything together. So it was a fantastic opportunity to learn how to present science, but also perform scientific experiments. And I think I can still remember my first one. We were testing vitamin C content in juices. And I sort of had a hypothesis that you could, as maybe a juice sat out, maybe the vitamin C content would change. So I think we had some orange juice and some grape juice that we put in sort of the refrigerator or just let them sit on the counter. And then I had a burette, and we were titrating in, I think, iodine that we bought at the drugstore to be able to sort of do these things. I think I measured everything in drops. A really scientific way of doing that at the time.
The metric drop, exactly.
Yeah, exactly. Yeah, I mean, I had the right tool to do it. I just don't think we had the skills to do it. And I think it was because my parents were so supportive of that, and I think they were super helpful in buying those materials. We still have the burette at home. I think they offered to donate it to my research lab, but unfortunately, I don't do a whole lot of titrations anymore. But I think that really paved the way for learning how to present science. And I remember the first time that I had gone through and did that presentation, I think I gave it to one of the teachers, and they're like, this isn't how you do this. Here's a better way to think about this. So I think that was a huge learning process. And I think I did experiments, putting preservatives in bread and looking at mold growth. I think we tried to kill some termites using some antibiotics for a lab. And I think I finally wrapped up with measuring like friction coefficients for tires on different surfaces. So spanning a huge set of things. Certainly nothing as complex as doing BioBuilder design. I think certainly there was a lot less biology that we had opportunities to do, but very exciting projects. And it was just great to see everybody who was doing it. And then, the thrill of discovery was there. And I think that kind of started my, pathway into sort of a research career.
I think so. I think it's just astonishing to me how clearly you remember these projects, right? I think there is such a lasting impact of working on a scientific or engineering question that is driven, that is self-directed, comes from something that as an individual you're curious about, and you can gather that data.
And those like aha moments when something works, it's just like, I think that's what really, it drives you forward. It drives you forward on those days when the experiments maybe don't work as much as you expect them to. But I think, putting it all together, it was just a great experience, and really, I think, provided a desire to do that for the career.
Yep. So then you went from high school to college knowing that you wanted to study science.
Yes. So I had done a lot of the research as a sort of high school student. I had done a bunch of engineering programs, I think, throughout high school as well, and kind of wanted to marry those two things together, which led to sort of even the choice of college. I wanted to go to a place where I would have the opportunity to do undergraduate research, right? So that was probably one of the primary deciding factors of where to go. I don't know that I necessarily knew exactly what I wanted to do. I think I was probably interested. I was probably going to be a mechanical engineer at most universities, but I was also interested in neuroscience at the time, and really just I knew that there was something in there that I wanted to do. And then I remember receiving a flyer in the mail about biomolecular engineering and thinking that that was really fun and interesting. I had never heard of it before. I knew I liked biology, right? Knew I liked chemistry, and then the mathematics was great. So I was like, let's give that a shot. And
Wow.
so I did that for my undergraduate and never looked back. So
Well, thank goodness for that flyer landing in your mailbox or wherever it was, because I think the awareness of all of these ways of combining the interests that students have, it's really not obvious. Students come in
no,
and they're like, well, I kind of like this and I'm kind of good at that, but I don't know what that adds up to. So the awareness raising is a very big part of what has to happen if we're going to bring all the talent along. Did you say where you went to undergraduate?
so I went to Johns Hopkins, so in Baltimore.
Yeah.
And I think it was a fortuitous choice because I think they had a lot of faculty who were doing this biomolecular engineering, people who were doing tissue culture engineering or cell mechanics engineering. Some other people were studying protein folding. I ended up working with my research advisor was studying allosteric enzymes. And so he had actually created an artificial allosteric enzyme in the laboratory. And I got the sort of opportunity to go through and sort of characterize some of this. We had discovered that it was actually partially inhibited by a metal, and we were trying to discover how it was inhibited by that metal. And then next thing you know, because we had made mutations, we were able to do some different surface science sort of measurements with it as well. And yeah, I liked it so much. I was there, I think, every summer doing research, and completed a master's degree as well. So I think it was one of those things where I got in really deep on it, spent a lot of time on the project, and it kind of led me to understand sort of what's happening in the field, right? Sort of where my interest level or different interests are, right? And then, certainly made me very excited to get a PhD. I think I probably went into college knowing that that's where I wanted to go, but I think I left college knowing that, I wanted to sort of go to that academic level after sort of my PhD.
That's very cool. It's not all that often as an undergraduate that people go so deep into a particular topic area. A lot of times people will touch on a bunch of different research areas, which is all good, right? So, but when you are in a place and it's the right thing for you to have that chance to really dig deep into the questions and make some headway, right? Because research does take a long time. It's not like you're going to find the answers in just a summer.
And now I get to see it on the other side, right? So I now get to mentor students in lab, and I think I get to see both sides of it, right? I see students who really do come in with a desire to go deep into a project, right? They really want to, make an impact, right? Understand things at a very deep level. And then I also have students who are, they really want to see what's out there, right? And they want to see the different areas, different projects, different ideas, different ways of thinking about research, right? So that they can make, maybe a more informed decision down the line as to which one they want. And I think both are really successful strategies. And it just depends on kind of, where you see yourself going.
Absolutely. Yeah. Yeah. I'm not surprised that you are such a dedicated mentor to your students and that you have the open sort of ideas about what they are there for and what their benefit could be. We share a friend and colleague in your graduate mentor. So do you want to take that next step and go from Hopkins into graduate school and tell people?
So I went to graduate school at Cornell. So I worked with Matt DeLisa there, and we were doing more protein engineering work. And that's where I actually learned about cellulase enzymes, which has helped me sort of transition those to those enzymes that degrade plastic, because they're actually fairly related to each other. And there we were looking at how can we make bacteria better at doing protein engineering. So we were coming up with clever ways for the bacteria to sort of know if a protein was well folded or not, so that we could better screen protein engineering libraries. So that was really exciting. But I also got to use a native secretion pathway. So we got to do a little bit of science there as well. Studying how that secretion pathway actually can sense a folded or unfolded protein is really interesting. We did secretion from cells as well. So we were looking at how can we make proteins leave E. coli, which is something that's, pretty hard to do. And we were coming up with ways of screening those. And it was just a great place and a lot of fun to see a bunch of different projects, right? And I think it has really allowed me to sort of when I've started continuing my career, seeing the different places that we can go. And I think one of the hallmarks that I take from that is learning from nature and sort of how we can use natural driven evolution, or how we can use nature's inspired strategies to really enhance what we're learning about. So if we go back to the fungal system, right, learning from the fungus that can already do what we're trying to do. I think nature has lots of years on us, even as biological engineering is taking off, right? I think we're still at an infancy there, and using those strategies is going to be really powerful.
Yeah, I appreciate how humble you are. I think sometimes people oversell this field and all that we can do, but I think that the notion that nature has a lot of years on us is spot on, because there are so many ways of solving problems and we have just scratched the surface.
Yeah, and I think that makes it fun for me as an academic engineer, right? So I'm in a chemical engineering department. I can see myself drawn to the engineering problems, right? So the application-driven science, but I'm also scientifically motivated, right? So I want to see those underlying mechanisms. And the goal is to find those kind of design rules so that when we go through and put the engineering design on paper, right, that we can improve upon that even further, right? And how can we understand these natural systems and what we can do with them? Because I mean, as we've seen from BioBuilder, there's many sort of problems out there that can be solved by these tools, these synthetic biology circuits, cells themselves, right? So how do we understand what's going on so that we can put forth the best solutions possible?
Yeah, that engineering process that overlays the scientific discovery is, I completely agree, how we're going to take this endeavor forward for sure. So, all right. So from Ithaca, where did you head after that?
Right, so that's when I moved to Cambridge and did a postdoc at MIT. And there we were looking at a different sort of native or natural system. So we were looking to culture bacteria under high pressures of carbon dioxide. And turns out biology doesn't like to exist under high pressures of carbon dioxide. Carbon dioxide can actually be quite toxic for natural systems, which is potentially a problem that we're running into as we keep sort of dumping more carbon dioxide into the atmosphere. So we isolated bacteria from an underground CO2 reservoir, and then my job was to engineer it. So it's this environmental isolate. And so we were able to make the bacteria fluoresce by introducing GFP, but then we were also able to make some biofuels and other bioproducts. And potentially this is a strategy for doing some different chemical processing. And it'll be interesting to see if this bacteria, right, can sort of do some other fun biology underneath sort of different pressures of carbon dioxide as we sort of move towards trying to find uses for this. So I think that's an exciting area that's going to be potentially very useful in the upcoming years.
Yeah, I think starting with that very important social question about how do we interact with CO2, and then to find the understanding that there are ways to engineer microbes to address things that are relevant in that process is vital. So how cool is that? Did you like your time in Cambridge?
I loved it. It was a great place to be. I think it was fantastic to meet everybody there. I think that's where I met Natalie as well as everybody else in BioBuilder. And it's a great community of scientists. I think between the different universities and all the pharmaceutical companies, all the startup companies, right, all the synthetic biology companies, right, I think there's just so much going on there that it's just this incubator for a lot of innovation, and very powerful. And I think that's what made it nice for all of the students as well through BioBuilder, is because they had that connection to somebody there. And I think it's very innovative, especially in this space. And I'm excited to see all the different clever solutions that people come up with.
Agreed. Yeah, there's a lot of cleverness in this, a lot of great innovation and ideas. Your backdrop is beautiful. Where did you land after being?
Yeah, so
that's not Cambridge.
That is not Cambridge. So I currently am an assistant professor at Miami University in Oxford, Ohio. And so I'm working in the Department of Chemical, Paper, and Biomedical Engineering. So keeping with that theme of chemical engineering throughout the process. Paper engineering is actually quite interesting, especially from a biotechnology standpoint, right? I think there are some people who are looking at enzymes to sort of do de-inking technologies. There's a potential for cellulases to play a role in recycling sort of paper and treating paper, as well as potentially even just designing better paper using protein. So I think there's some fun aspects there that I'd never even thought of before coming here. But so Miami, being a public liberal arts sort of school, we're focused on undergraduate mentorship, and sort of that was what drew me to this program, right, was the sort of ability to work with students at the bench and really train the sort of students who are going to go to graduate school or are going to get those R&D jobs, and really start at the sort of beginning of their research career and being able to influence them there. So I think that's been a lot of fun for me. It's certainly one of those things where, you know, I have now my own sort of research projects that are ongoing, and we're looking at transcription factors. We have some other projects working with industry on different laundry detergents, as well as, you know, different biomaterials using elastin-like polypeptides. So we have a lot of sort of space that our lab has started going into already, which is really exciting. And you know, it's a fun environment to be in. We're very collaborative, so I think that's helpful. And you know, certainly the students are getting a fantastic experience. I'm looking forward to them getting out and presenting some of this research soon.
It's amazing. I had not thought about paper engineering, and as you say, there is so much. I love that it is a collaborative endeavor with chemical engineering and medical engineering. It sounds so smart, and I do think that's how you're going to solve these intractable, very difficult problems, is by working across boundaries that you wouldn't normally think, you know, could.
Yeah, I've found that I think the most successful collaborations are the people, if you're speaking exactly the same language, it's hard to sort of have a really meaningful collaboration. It's really when you can bring people who have slightly altered skill sets, right, and sort of put everybody together into that sort of brainstorming session to come out with, you know, how can we really be innovative together? And I think that's where science is happening right now, is at these interfaces. And we're really sort of starting to break down these sort of disparateness between, say, even science and engineering, right? I think there's a huge amount of overlap where we're different fields within science or engineering, right? We're collaborating with each other. So I think thinking about this is really, how can you impact things by putting together your own training, I think is very important.
I wish someone had said that to me when I was back in high school too. The notion that you had to choose where you wanted to work, you know, it really runs counter to that idea that having some cross training, having some vocabulary that you can share with people who have different focuses, but there aren't walls between these different disciplines. It's really much more helpful when you can have conversations across them, for sure.
Yeah. And even across, like, you got to talk to the business people too. So there's, you know, you have to be able to talk to everybody about things. And I think, you know, if there was one thing that I sort of, you know, learned through this from high school, is how big communication is for this whole process. And I think I was lucky enough to participate in a program called GK12 during grad school, where I got to go to high schools and actually share sort of some laboratories and develop some sort of science for them. But, you know, it really put forth this idea that, you know, as a scientist, I thought, you know, you go into the laboratory and, right, you create a great experiment and you put it in a laboratory notebook. But if you're not great at science communication, that's all the further it ever goes. It never leaves the laboratory notebook, right? And really, it's sharing that with your peers, right, in science, but it's also sharing it with the general community. And how can you sort of bridge those different audiences and make sure that you're targeting, or you're talking about those things to people, I think is really important, right? Because that goes back to your underlying motivation for doing the work of benefiting society, right? And if you can't talk about it, then it's really going to be hard to say that that's a useful project or a useful endeavor to go through. So that's what I would tell people, is I end up spending much more of my time writing and talking than I probably would have ever thought in high school. I thought it was all just this bench work, and that's just not what it is. It's talking to people about the science and what you're doing.
Ah, what great advice. And how lucky are we to have you in the BioBuilder community, because sharing the work that we do is just wrapped up in it, right? We do a lot of team-based things, we do a lot of communication and celebration of the work. So boy, are we lucky to have you in it. And those students are going to do great things with your mentorship. Congratulations on all the success. I'm
Oh,
so
well, thank you.
excited to
Yeah.
hear about
And
it.
you guys as well. I mean, it's been so fun to be part of the community for the past 3 years and just get to meet all of the different students as well as everybody associated with BioBuilder, and the excitement that they bring is fantastic to all of these events.